left out histology portion kasi si Doc Lasaka ang bahala sa inyo niyan. On a broad scope, it is also known as your leukocytes. In lieu with your RBCs, erythrocytes, pag makarinig kayo ng leukocytes, ang pinapertain niya, in general, please take note, your WBCs. It can either directly attack the pathogens, it can be mediators and key signal providers. Nagproproduce sila ng mga interleukins, mga inflammatory proteins, or it can produce your antibody. It can produce your antibodies. That is your lymphocytes, a form of your leukocyte. Generally divided into segmenters and non-segmenters. When we say segmenters, yan yung neutrophils, eosinophils, and basophils. Non-segmenters, those are your monocytes and your lymphocytes. And it is a key participant in your acute inflammatory response. These are the steps of your acute inflammatory response. Please take note of this. First reaction in your acute inflammatory response, it is increase in vascular permeability. Once na mataas na yung permeability ng blood vessels mo, the WBCs from the blood vessel, makakalusot na siya from the blood vessel, and it can go to your tissues. Then it will activate the platelets, then resolution. Please take note of these steps. Increase in vascular permeability, entry of WBCs in the tissues, activation of the platelets, and resolution. So this is the maturation of your WBCs. Kanina, stem cell, nakita naman natin, stem cell divided into a erythrocyte precursor, a myeloid precursor, and a lymphoid precursor. Dito, when we talk about WBCs, dadalawa yung kanyang precursor niya, your myeloid stem cell and your lymphoid stem cell. Your myeloid stem cell can produce your platelets, your monocytes, your neutrophils, eo, and baso, as well as your erythrocytes, with the help of your erythropoietin. Tapos si lymphoid stem cells, separate lang siya para maging lymphocyte. Meron kasi isang lymphocyte, your B lymphocyte, nag-ano siya sa thymus, nagmamature, bursa or fabricius. Isa yan sa area niya. Pero anyways, i-discuss niya sa inyo histology. Sa akin yung biochemistry portion. There are so-called memory lymphocytes. When we say memory lymphocytes, it can either be a memory T cell or a memory B cell. The memory is produced when an antigen is presented to them. Okay, please take note of that. A memory is produced when an antigen of a specific bacteria, virus, or fungi is presented to them. Anong gamit ng memory T cell and B cell sa katawan? Once na parang babae ito kasi sila actually, ano lang to, na mga historical, yung madaling makaalala ng mga pagkakamali, parang ganyan yan sila. Once na merong yung pagkakamali dito, yung mga bacteria and viruses, once na bumalik ulit yan, pag makita nila, madali lang remember, madali silang makaka-act on it. That is the function of your memory T cells and B cells. Okay, they can act immediately on the second time na i-present sa kanila yung infection. So, nangyayari ito pag meron kang AIDS or HIV infection, nasisira yung T cells mo, bumababa yung memory lymphocytes mo, kahit paulit-ulit ka nang na-infect ng virus or bacteria sa HIV, it's as good as new kasi hindi siya nakaka-form ng T-cell, memory T-cell or memory B-cell dun sa HIV. Kasi kulang na, mababa na sila yung concentration. Now, production of the cells can be stimulated by different cytokines. Okay? Mamaya we will talk about those cytokines. These are proteins that stimulate the formation or reaction of the cells. So we have the movement called chemotaxis. It is the migration to a target location via chemical signal. This is the step number 2 in your acute inflammatory response. Given na increased vascular permeability na, ito yung capillary endothelium mo, if there is increased vascular permeability, yung chemokines mo would be released to your blood vessel. Your chemokines are signaling proteins na nagsasabi sa mga WBCs na andito kami, andito ang infection, andito kayo dapat pumunta. So once the chemokines is received by your WBCs, it already knows where to go. Okay? So papasok yan sila sa area na may infection with the movement called diapedesis. So this one, ito pag nandito na siya sa tissue na yan, that is your diapedesis. Please take note of that. So these are the form of yours, chemokines. We have 4 subclasses. Okay, the most common is the C chemokine, this one. Tapos yung difference lang ng mga subclasses nila is this one. This area here. Sorry. This area here. The branch amino acid, the in-between amino acids, okay? The in-between amino acids, that X. Yan lang yung nagkakaroon ng difference yung chemokines mo. Pero the most important here is it is stabilized by a disulfide bond between 2 cysteines. This is your disulfide bond on 2 cysteines. This one stabilizes your chemokine. Kasi pag wala to sila, mag-unfold yung chemokine mo, hindi na siya magiging effective. Okay? Then we have also the so-called integrins. Integrins is a transmembrane glycoprotein that facilitates, please take note, of adhesion of your leukocytes. Okay? Nakikita nyo to dito. For this WBC to adhere on the capillary endothelium, it needs integrins, okay? It integrates your WBC to your capillary endothelium. And once it binds, meron na tayong tinatawag na rolling in na maneuver. Then diapedesis, papasok na siya sa tissues. Pero it will never be able to go inside the tissue or pass through the capillary endothelium kung walang integrins. Okay? Kasi, please take note, this is a flowing medium. Tuloy-tuloy yung pag-flow nito. Every pump of your blood, gumagalaw to siya. Okay? So, isipin mo na lang, wala kang anchor, para kang bangkaw na walang anchor sa port, lulutang at lulutang ka niyan. So ang function ng integrin, it will bind your WBC to your capillary endothelium. So it has an alpha and beta subunit. And the extracellular unit binds to any protein that has the, please take note, very specific, arginine, glycine, and asparagine sequence, or aspartate sequence. Okay, so it will bind to the external protein of your WBCs with this sequence, arginine, glycine, aspartate. And the intracellular is bound to the cytoskeleton of your endothelium. The main function of your WBCs, one of the main function of your WBCs, phagocytosis, or the engulfment of a non-self matter. When we say non-self matter, that is your bacteria, viruses, and your mga fungi. It can bind to a target cell by their own structure or recognized indirectly via adherence of a protective protein, or the so-called process of opsonization. When we say opsonization, there is a certain protein like your antibodies na parang ibabind niya yung bacteria, and it will signal to your WBCs na ito non-self to kasi nakabind sa antibody. So the WBCs will react, then i-engulf na niya. That is the process of your opsonization. Pero there are times na hindi na kailangan ng opsonin yung bacteria or any antigen. Okay? It depends on the infectivity or the antigenicity of your non-self matter. Kung highly antigenic siya, madali lang siyang madidetect ng WBC mo. Hindi na kailangan ng opsonin. Pero pag low antigenic siya, highly virulent but low antigenicity, yun yung mga delikadong bacteria natin. Andyan yung mga antibodies mo, isosurround siya, i-opsonize siya, tapos matidetect na ng WBCs mo na, yan, delikado yan. Kasi na palibutan na ng opsonin. So, i-engulf na siya, sisirain. Engulfed cell is destroyed inside the phagosome, wherein enzymes from the granules enter. This is a very important concept. The phagosome is a controlled environment. Dapat nandyan lang siya sa loob, and lahat ng enzymes na sisira sa bacteria, andyan lang yan sa loob. Kasi once na mag-leak yung enzymes mo sa loob, palabas ng phagosome, cell destruction, tissue destruction yung mangyayari. So, that phagosome is a very controlled and very intact area. Okay? The debris are a source of antigen that can be stimulus for production of new antibodies. Yung sinasabi ko sa inyo na na memory T cells and B cells, andyan na yan ito na yan sila. Ito na yung function nila. Nagkakaroon ka ng memory T cell and memory B cell dahil dito. Yung antigen presentation with the help of your major histocompatibility complex. Okay? Your helper T cells and other leukocytes present this on their surface along, please take note, your major histocompatibility complex for antibody-forming lymphocytes, your B cells, your memory B cells and T cells. Okay? Hindi siya ma-acknowledge na antigen or hindi siya ma-acknowledge na kailangan tong gawan ng antibody kung wala si major histocompatibility complex. Please take note. Para lang siyang food panda nagde-deliver na naka-t-shirt kung walang major histocompatibility complex. Si major histocompatibility complex, yan yung kanyang uniform na color pink. Na ito, delivery boy ito ng food panda. Parang ganun yung analogy niya. So it will never be able to be recognized na antigen siya na kailangan gawa ng antibody kung wala yung major histocompatibility complex. Kaya pag may abnormality yung tao na kulang or abnormal yung major histocompatibility complex na protein. This is a protein kasi your MHC. Pag walang MHC or kulang ng MHC, walang antibody or kulang yung antibody na maproproduce. So, same as to your HIV problem or any chronic infections, kahit paulit-ulit kang ma-infect, walang nagagawang memory, lagi ka lang magkakaroon ng flu, magkakaroon ng cough, kakaroon ng fever with the same bacteria or virus. So that is the importance of your major histocompatibility complex. As you go on with your study in medicine, meron pa yan MHC1, MHC2. Kung medtech ka, mas elaborated yung study mo nito actually. Kasi marami pa yung paikot-ikot sa MHC. I will not discuss anymore yung paikot-ikot ng MHC yan. Pero all I want to emphasize is that this is a very important protein for antibody production. Okay? So that it will recognize the antigen na kailangan gawa ng antibody. So there are 3 general phagocytes. Please take note: neutrophils, eosinophils, and monocytes. Those are your phagocytic WBCs. So this is the general steps of your phagocytosis. Step 1, being identified as a pathogen based on its own protein. Wala tong opsonin, ha? Wala tong opsonin. Now, when it is recognized, it will bind. Step number 2. Then it will be engulfed or phagocytized. This is the act of phagocytism. Ito. Then, yan, once the engulf, that area there, that enclosed area there is your phagosome. Now, once inside the granules of your WBCs, it will attach to your phagolysosome or your phagosome, and it will form your phagolysosome. Sa granules, maraming enzymes yan na for degradation. Okay? Now, once degraded na yung bacteria or any pathogen, your major histocompatibility complex, it will attach to one of the proteins. Okay? Once attached, i-represent niya sa labas to your T cells and B cells. Tapos, gagawa na sila ng antibody for that pathogen. So this is very important, ha? Phagocytosis. Kasi pag walang phagocytosis, pag hindi niya nasisira to, hindi siya makakaproduce ng protein na ibabind sa major histocompatibility complex. Tapos, wala siyang i-represent, walang memory. Okay? Walang memory na magagawa yung katawan mo. So, recurrent infections ka. Yan yung common signs and symptoms ng mga immunologic patients, immunodeficiency patients, recurrent infection. Kasi ito siya. One of the parts yan. Yung iba naman na walang MHC, once na ma-degrade, ini-excrete na lang yung degradatory protein. Sinisira na lang siya sa spleen. So please, these are the enzymes inside the granules of your WBCs. You have your myeloperoxidase and your NADPH oxidase. Those two are the most common enzymes inside your WBCs because those two are the ones that produces your reactive oxygen species, your ROS, that destroys the bacteria or the pathogen And the other one stabilizes your reactive oxygen species. So your NADPH oxidase produces your ROS, then your myeloperoxidase stabilizes your ROS. So, kanina meron tayong cytochrome system sa RBC. Ang gamit niya ng cytochrome system doon is to convert the oxygenated iron o oxidized iron or your Fe3 plus to Fe2 plus. Dito naman, the most important system in your WBC is your NADPH oxidase system. Okay? Huwag niyong babalik-balik ta rin yung dalawa ha. NADPH oxidase system is the one responsible in producing your reactive oxygen species and initially stabilizing those reactive oxygen species. It all happens inside the phagosome. So, sinabi ko kanina na kailangan ma-contain talaga yung phagosome na area kasi doon lumalabas yung reactive oxygen species mo. Your reactive oxygen species are the free radicals being produced by your NADPH oxidase system, the so-called respiratory burst. Okay. First, divided into two. The first step is the reduction of your molecular oxygen to form your superoxide. So, four molecules of oxygen with the help of your NADPH and then and hydrogen will form your superoxide. This is your superoxide. It will remove an electron, okay? That makes your oxygen unstable and will produce your superoxide. That superoxide will act on the bacteria, okay, oxidizing the proteins in the bacteria or any pathogen and destroying it, okay? That's the function of your superoxide. Now, simultaneously, mabilis lang yan na reaction. Pag masira na niya yung bacteria, yung virus, kailangan siyang ma-stabilize. Kasi pag hindi siya ma-stabilize, pwede niyang masira yung phagosome, pwede siyang lumabas sa phagosome, masisira niya yung WBC. The second step is the neutralization of your superoxide. Okay? The superoxide will bind to your hydrogen ions to produce hydrogen peroxide. This is the final product of your NADPH oxidase system, your hydrogen peroxide. So this is a very, very fast reaction inside the phagosome of your WBC. Now, when you have a bleach or your hydrogen peroxide, the myeloperoxidase will produce a acid on it. Okay, to neutralize the hydrogen peroxide. Ang usually lumalabas nila hypochlorite, HOCl, then water. Then stabilize na yung hydrogen peroxide mo. Okay? So your NADPH oxidase and your myeloperoxidase go hand in hand. Your NADPH oxidase produce your reactive oxygen species. Then your myeloperoxidase produces hypholus acid plus water. Now, anong mangyayari kung walang NADPH oxidase system? Nung med school kami, I always tell this to my class, sa barkada ko, na pag wala kang NADPH oxidase system, para kang always on the third base. Sa mga lalaki, always kang third base or always kang friend zone. Makaka-produce ka ng phagosome, walang problema. Third base ka na. Pero hindi ka makaka-go all the way kasi walang makaka-produce ng reactive oxygen species mo na superoxide. So hindi masisira yung bacteria mo. And it will form tawag niyan, granules or granulomas in the body. This is the so-called chronic granulomatous disease. Please take note of this disease kasi favorite question to ng pathology professors pag mag-second year na kayo. Your chronic granulomatous disease, your chronic granulomatous disease is a genetic disease wherein your WBCs cannot produce superoxide because wala siyang NADPH oxidase. Okay? Wala siya yung enzyme na yun. Wala siyang NADPH oxidase. So please take note of that. Your chronic granulomatous disease, deficiency of your NADPH oxidase, no formation of your superoxide, recurrent infections, productions of granuloma in the body. Okay? Huwag niyong i-interchange to kay kay cytochrome B5 reductase system. Anay daw. Dane?
Yes, Doc.
Okay. Oh, good man na, nag-enroll ka ngayon.
Ayaw na.
See, anyways, now, we're done with the viruses, the bacteria, on reaction of your WBCs. Let's go naman to your parasitic response of your WBCs. Very unique. Neutrophil extracellular traps or nets. These nets or your neutrophil extracellular traps, please take note, not a protein, not a carbohydrate, not a lipid, but a DNA. These are strands of the DNA of your WBCs na once na masira yung WBC mo, na nagfo-form niya as the moniker nets, and it will entrap your parasites. Okay? So how is it formed? So the strands of the polynucleotide from the neutrophils, chromosomal, take note, DNA, is nadideaminase yung arginine portion ng DNA to your citrulline. Okay? Please take note of that. Arginine portion, it is converted to your citrulline in the polynucleotide. Now, once citrullinized, the so-called citrullinization, the neutrophils, given na sira na yung kanilang DNA, kasi yun na nga, yung arginine na convert na to citrulline, masisira sila, it would lyse. Now, once lysed, those DNA na naging strand because na citrullinized yung arginine to citrulline, they would form this one. Ito siya. It will not be dissolved because citrullinated na siya. Okay? Thus, it would bind to your parasites. So, yan lang yung function niya. Hindi niya papatayin yung parasite mo. Iba yung papatay sa parasite mo, your eosinophils. Yung gamit lang niya is to entrap the parasite. Okay? All I want you to remember is the citrullinization process. That the arginine is converted to citrulline so that your polynucleotide would become strands. Okay? Then the granules pala. yung granules ng WBCs nyo, kasi isang term pa ng mga WBCs granulocytes, kasi meron silang maliliit na granules. Once na mag-ano kayo sa histology, makikita nyo na may granules yan ng mga WBCs na yan. Yung neutrophil, eosinophil, and basophils, they have granules. And each of them has a specific enzyme inside it. So those enzymes, dito sa nets, mabind rin siya sa dun sa mga strands. Okay? To further immobilization, ha? Pero ang kanyang main function ng nets mo is immobilize lang. Please take note of that. This is the reaction of citrullinization. Your amine here is being replaced by oxygen. Okay? With a byproduct of ammonia. Or ammonium. Ammonium to kasi ion. That is your citrullinization. Next is your eosinophils. Main WBC, please take note of this, main WBC that acts on your parasites. Kung marami kang bulatis sa tiyan, mataas yung eosinophils mo nito. Okay? So, kung sa physical exam ninyo, mataas yung eosinophils ninyo sa differential count, alam nyo na gagawin. Purga na lang. So, the eosinophils has a very, very important, the major basic protein sa kanilang granules. That is their main anti-helminthic protein. Okay? That is their main anti-helminthic protein, your major basic protein. Major pero basic. Major basic protein. Their function is the MVP. Ang mangyayari niyan, it will attach to the surface of your parasite and create pores. Okay? Bubutasin nila yan. Yung surface ng helminth mo. And the other enzymes will follow na. Okay? Then madedegrade na yung parasites mo. That's the function of your eosinophil. Primary, antiparasitic, WBC, eosinophils. Please take note of that. Now, yung kanina kong sinabi, cytokines, these are your small secreted proteins mainly used in communication and coordination of complex immune inflammatory response. So, pag may problema sa cytokines mo, disarray yung immune system mo. Hindi alam ng mga components kung anong gagawin nila. Ano bang gagawin ko? May sugatan ka. Pag wala kang cytokines, hindi mo alam kung anong gagawin mo dyan. Okay? That's the function of your cytokines. This is the common sense of your immune system. So it is divided into 3, interleukins, interferons, and chemokines. Interleukins, marami yan. Interleukin 1 to interleukin 12. Meron ng iba actually. Pero yan yung nare-remember ko. Interleukin 1 to interleukin 12. Interleukin 1, fever and inflammation, as well as 2. Interleukin 3, 6, and 3, 4, and 6. 3, 6, and 9. Yung kanina. Yung maturity ng stem cells. And so on and so forth. Sobrang rami yan nila ng mga interleukins. I'm not saying na kailangan nyo ma-memorize lahat. Plus, just please take note of your key interleukins. Like interleukin 1, interleukin 2, 3, 6, and 9. Now, interferons. As the name suggests, its main function is to inhibit. Okay? Inhibit viral infected cells. Okay, that is the function of your interferons. There are 2 kinds of interferons, but the main function of it is to inhibit. Then, the third kind is your chemokines. Na-discuss ko naman kanina yung chemokines. 4 subclasses, uniquely stabilized by sulfide bond between two cysteine. Okay, most common is the C subtype, and its function is to attract your WBCs to the site of injury. Okay? Your cytokines can function as an autocrine, self, activity, and paracrine to the others. Your chemokines, it suggests paracrine talaga siya. Tissue, mga damaged tissues, magproduce ng chemokines, i-attract ang WBC. That is paracrine signal. Now, histamine. Major WBC that contains histamine, please take note, basophils and mast cells. Okay, basophils and mast cells produces your histamine. And histamine is a product of the decarboxylation of your histidine. Please take note of the reaction, decarboxylation of histidine. And the main reaction of histamine is promote edema. Kaya, pag nagkakaroon ng allergic reaction, edematous yung airway or namamaga yung airway because of release of histamine. Kaya magbibigay sa inyo sa emergency room ng diphenhydramine immediately kasi some allergic reactions are lethal kasi. So diphenhydramine, antihistamine yan. Pero on the note of biochemistry, please just take note, decarboxylation of histidine promotes histamine. Ito yung kanilang mga classification, mga actions. Autocrine, self, paracrine, and endocrine. Paracrine and endocrine are different because paracrine, nearby cell lang, like one WBC to another WBC. Pero mga endocrine mo, action, distant cell. Like your thyroid glands to other organs, or your like kidneys producing erythropoietin. Okay? As I was saying kanina, interleukin 1, please take note, fever. It is the main cytokine for your fever. Kanina, 3, 6, and 9, that stimulates your bone marrow. Okay? Pero 4, 5, mga B cells. Good to, I'm not asking you to memorize this, ha? I just want you to take note yung favorite question, what interleukin promotes fever? Interleukin 1. Hindi nyo na yan makakalimutan. Hopefully. So, inside the WBCs, there are also eicosanoids. Secreted lipid mediators, a product, please take note, arachidonic acid oxidation. Na-discuss na to ni Dr. Monsanto, wala pa. Din. Na-discuss na, wala pa. Anyways, very in-depth yung discussion ninyo to. I will broadly discuss lang to. Your eicosanoids has two categories, your leukotrienes and your prostaglandins. Please take note, your leukotrienes has three carbon-to-carbon double bonds, as well as incorporated cysteine in their structure. Okay? No rings sa leukotrienes. Leukotrienes, 3 carbon-to-carbon double bonds with a cysteine in their structure. Pero ang prostaglandin, 20 carbon atom with 5-member ring. So kung tanungin sa inyo or pakita sa inyo what eicosanoid is this, tapos makakita kayo ng 5 carbon ring, prostaglandin na kagad yan. Pero kung makakita kayo ng 3 double bond na carbon to carbon, leukotrienes yan. So essential fatty acid, your omega-6 and fatty acid would be converted to your arachidonic acid. Then can be acted by 3 enzymes. Lipooxygenase that produces your leukotrienes, cyclooxygenase that can produce your prostaglandins and thromboxane, and cytochrome P4 or 50. Okay? Ito yung dalawang mas importante. Okay? Your lipoxygenase and cyclooxygenase. Mefenamic mo, ito yung ini-inhibit. Cyclooxygenase. Tapos yung cyclooxygenase mo, 1 and 2 yan. May type 1, may type 2. Depende sa tissue area mo. Okay? Ang ginagamit na ngayon, ini-endorse ng karamihang doktor ay your cyclooxygenase 2 inhibitors. Kasi tawag niyan, hindi nakaka-produce ng ulcer. Okay? Hindi nakakasira ng gastric lining. Kasi ang present sa gastric lining mo is cyclooxygenase 1. So, the functions, leukotrienes, vascular permeability, lipoxins, anti-inflammatory, prostaglandins, inflammation and vasodilation, thromboxane, hemostasis. This we will talk about sa next lecture natin sa hemostasis. So, yung sinasabi kong decarboxylation, when you say decarboxylation of histidine, tumatanggal ka lang ng carbon dioxide. Now, we've talked about granulocytes or your tawag niyan, segmenters, neutrophils, eosinophils, and basophils. Let's talk about lymphocytes because they both have their own functions, and lymphocyte, its main function is the adaptive immune response. It is the cornerstone of the adaptive immune response. So, kaya nga nangyayari sa HIV, recurrent infections kasi kulang na ng lymphocytes, mainly T lymphocytes. The B lymphocytes secrete soluble antibodies. Na-discuss na natin yan. That is the so-called humoral immunity. Ang question is, what is the trigger of synthesis of antibodies? It can be direct binding to the pathogen via cytotoxic T cell. Oh no, iba to. Direct binding to the pathogen and to your antigen-presenting cells with MHC or your major histocompatibility complex. Yung sinasabi ko kanina, this is your granulocyte. Or sabihin na natin tong, neutrophil. Si neutrophil, nakakain ng microbe, tapos na niyang ma-degrade sa kanyang phagolysosome with your NADPH oxidase system. Release na niya yung debris. Tapos, kinabit niya yung isang debris or yung isang antigen fragment to your MHC, this one, and it presented itself to your surface. Then, ito siya. 2 kinds. Cytotoxic T cell, helper T cell. Si helper T cell, ang kanya lang gagawin is it would encourage antibody production through activating your B lymphocytes. Si cytotoxic T cell, ang gagawin niya, ito siya is an infected cell. Yung difference ang nagbabind kay cytotoxic T cell, isang infected cell natin, for example, is a cell sa ating intestine na infect ng isang bacteria like salmonella. Now, na-degrade na siya. I-represent siya inside via class 1 MHC molecule to your cytotoxic T cell. Itong cytotoxic T cell, papatayin to siya na infected cell. Okay? That's the difference between cytotoxic T cell and helper T cell. Helper T cell for long-term reaction, cytotoxic T cell for short-term reaction. Okay? Nakuha tong concept na to, yung helper T cell and cytotoxic T cell, and the class 2 and class 1. Kasi ito yung specificity ng MHC. Your class 1 will bind to your cytotoxic T cell, your class 2 to your helper T cell. Your cytotoxic T cell, given the name cytotoxic, will kill the infected cell, while your helper T cell will promote antibody production and aid in the infection. Pero hindi siya mismo ang papatay sa bacteria.